Good afternoon. It gives me great pleasure to welcome you at the first expert lecture at NAS. That is the Faculty of Natural and Agricultural Sciences at the University of Pretoria. The purpose of these lectures is to showcase our best and brightest and and bring their minds to bear on the cutting-edge problems of the day. It gives me great pleasure to introduce to you today Professor Thulani Makhalanyane. Professor Makhalanyane managed a master's degree from the University of the Western Cape, cum laude, as well as a PhD. And then he joined the University of Pretoria at 2012. And then he started a postdoc in 2013, looking at the microbial ecology in our desert biome. And from there, his career has really launched in a very, very impressive trajectory. He received an award for service excellence from the International Society of Microbial Ecology, and he also spearheaded a bid to host the first International Society for Microbial Ecology meeting on the African continent in Cape Town 2020. Well, we all know what happened in 2020, but I do know that the planning for this conference is ongoing around the pandemic restrictions. Professor Makhalanyane is also recently the South African lead on an international project called Atlant-Eco, and the focus here is on the role of the microbiomics in the ocean. From my own research in climate change, I know that there are climate modelers that have modeled that up to 40% of the global carbon cycle might be coming from the Southern Ocean. But we really do not know the mechanisms at all, and the research that Professor Makhalanyane will be leading, already started, will tell us a lot more about carbon sequestration and other ecosystem services from this very, very important part of our globe. Without further ado, Professor Makhalanyane, I look forward to your talk entitled Leapfrogging Innovation in Africa by Harnessing the Microbiome.
Thanks, Prof. Erasmus, for that very nice introduction. Many thanks as well for the opportunity to share some of our work on the microbiome. I hope that by the end of this talk, you will all be as enthusiastic as I am about applying microbiomics in your specific field of research. I will start by providing an overview of the field. Then I'll talk a little bit about some of my research. I won't go into detail of the research that we've done. And then I'll conclude by making a case for how microbiomics can leapfrog innovation on the African continent. That's right. So my talk will have an introduction, it will have a middle section, and it will have a conclusion. So I'll start first by acknowledging the people that are shown in this slide. And that's not everyone that I should acknowledge, but I think this is a good point to start because the work that I work on and that others do is not really an effort from my own individual efforts, but I owe it to all the collaborators, both internationally and nationally, who help and contribute towards our work. So I'm part of the Center for Microbial Ecology and Genomics. We currently have 3 staff members. We've got over 50 students that are linked at various capacities in our group. Our fields of interest span these 4 areas. We're interested in understanding microbial processes, that's looking at adaptations and potential applications. That's work that relies a lot on trying to understand bioprospecting for new enzymes, stress response genes, and so on. We are focused on understanding the microbial ecology of extreme and local environments, and we're also interested in looking at host-associated microbiomes, and I'll speak a little bit about some of these. We are interested in agricultural microbiomics, and there's also a strong team that looks at plant pathology within our group. I'm also quite interested in understanding the microbiomes of geographically strategic marine environments, and I'll talk a little bit about why I'm interested in these. So my current projects through the microbiomics at UP Group are the following. We're interested in a project that is focused on providing an understanding of the ecology, evolution, and function of marine microbiomes. We have a project that is currently looking at Antarctic permafrost, looking at the effects of warming on these systems and potentially what new microbes may come out from soils that have remained frozen for millions of years. We're interested in understanding the microbial ecology of South African soils. And this research spans from soils that are very hyper-arid to soils that are found in more mesic environments. We've got a new project where we're trying to look at viral diversity in aquatic systems that are linked to the water that we take in. Specifically, we are looking at what are known as crassphage genomes within these waters. Another project that we do in our group focuses on using human or understanding how living in urban and rural settings affects the gut microbiomes of humans in South Africa. So I know you all came for a science talk, but So if you were to take the Earth's 4.5 billion years and you would compress it in a 24-hour cycle, humans would first arise in the last 40 seconds. The first evidence of unicellular life would appear if you were to look at the 4.5 billion years in a 12-month cycle. The first evidence of life would appear in February. Multicellular organisms that are closely related to us would then come about in August. And what you can see from this slide and what's displayed here is the centrality of our microbial companions and how they've been here for much longer than we have, and how their actions have actually brought about the world that we live in. You would not be here if microbes were not here. So there are key moments that have defined the field that we're in. I've just shown the past 300 years or so and how the field of microbiomics has developed. There have been very instrumental developments which allowed us to first get the ability to visualize microorganisms. Of course, most of you would know that microbes are invisible to the naked eye. So being able to study microorganisms required the development of technologies which would allow us to amplify so that we can see them with our visible eye. There's been other developments in the so-called golden era of microbiology, led by Louis Pasteur and Robert Koch, which have led to providing fundamental insights of microorganisms using culture-based methods. So what are culture-based methods? A lot of the work that's been done up until 30 years ago relied on us being able to first grow microorganisms in a laboratory setting. So with these methods, we were able to only understand and grow 1% of the microorganisms that are out there. It is only up until the late 90s where we were able to get access to the 99% of microorganisms that cannot be grown in the lab. So there's various reasons why these microorganisms cannot be grown in the lab. So imagine you are looking at microbes that are sourced from 5 kilometers deep underground or under the sea. We're producing those pressures, the micronutrient conditions, and all the different aspects that they require for growth is almost an impossible task. The development of what are called microbiomic approaches in the late 90s allows us access to all the organisms that cannot be grown in the lab. So this slide here illustrates a little bit about some of the technical developments that have gone on. So you can see on the slide displayed that you have a mixed microbial community. So if you were to pick up a grain of soil from the ground, that grain of soil would have up to billions of different microorganisms. So there's a technical challenge in then trying to first understand exactly what those billions of microorganisms are. Microbiomics and so-called culture-independent approaches have allowed us to be able to extract the DNA or the nucleic acid content of those organisms and be able to study them in the lab. So there are two different approaches. So as you can see, on the top of the slide are what are called amplicon-based approaches. So these are the approaches which allow us to select genes of interest, right? So fragments of DNA that we are interested in, and be able to use these to get an understanding of which microbes can be found in any kind of environment. So these are called amplicon-based approaches. So there are other approaches that we can also use to study microorganisms. So these are so-called shotgun metagenomic approaches, and these are approaches which essentially allow you not only to look at discrete gene fragments, but to look at the entire community that's present in any environment. So the challenge of both of these methods is that you are amplifying, or if you're using shotgun approaches, sequencing DNA from an entire mixed community, right? So this places a computational burden, right? You can imagine if you have a puzzle which comprises of 20,000 pieces, and trying to put back these pieces together requires a level of organization to be able to get to a point where you know exactly what the puzzle represents. And that's a perfect analogy for trying to carry out what are called shotgun metagenomic approaches. So both of these approaches allow us to know first who is present in any type of environment, and also give us information about what these organisms do in these environments. So what's shown here is our current view or understanding of the phylogenetic tree of life. Phylogenetic is a big word, but essentially what this shows, it's a tree. And on this tree, the different colors and the different dots that are displayed there represent all the organisms, the groups of organisms that are known to life. At the bottom of the slide, in green, would be where we belong as human beings. And our ancestors that are closely related would also fall in under that group. Things like plants and so on would also fall under what are termed eukaryotes. What you can see from this tree is that the vast majority of the organisms that are present are actually microbial. So microorganisms outnumber us many million folds over. And these microorganisms, as I mentioned earlier, are highly abundant in any type of environment that you could look at. So the example is often made that if you take a bucket of seawater, a little drop of that seawater would contain hundreds of millions of microorganisms. So these microorganisms are not only there, but they actually carry out very important functions. We know that, as Professor Erasmus mentioned, that the oceans sequester or cycle a lot of the carbon dioxide that's produced. Microorganisms are actually the drivers of those ecosystem services. And our group and others are trying to understand precisely how they carry out these processes. But there are lots of complexities, right, that are associated with trying to understand exactly what microorganisms do. The first I've mentioned is that they are highly abundant. The second is that all of these microorganisms, irrespective of the environment that you look at them from, they are always in a state where they are constantly interacting with each other. This interaction means that all the microorganisms that are close to each other may be sharing genes, they may be losing and gaining some functions, and this makes it very difficult for us to get a clear idea of which microorganisms actually perform a particular function. This complexity also makes it difficult for us to get a clear sense of how microorganisms may vary across different ecosystems. But there are some insights that we've been able to gain, right? So I've told you that microorganisms are quite important in carrying out things like carbon cycling. We know that microorganisms provide other important services. For instance, plants would not be able to produce or photosynthesize without the ability of the nitrogen, which is gained through microbial actions. We know that in our bodies, for instance, microorganisms help break down compounds that our bodies can simply not do. So microbial communities are highly important, and I'm hoping this slide gives you just a clear approximation of just how important they are. So in preparing for this talk, I looked at the state of microbiomics globally. I did a simple Scopus search where I looked at the search term bacteria. And what you can see on the left of this slide is that there are over 70,000 publications that have been published in 2020. These publications span a number of different fields. So everything from medicine, biochemistry, immunology, material sciences, engineering, and even the social sciences. So there's an application of microbiomics. And this slide also shows the multidisciplinary nature of the field. So we've gained some insights from all of these over 60,000 publications plus about the role that microorganisms play in host-associated systems. That's microbes that are linked to us as human beings. We know what microorganisms do in soils. So things like breaking down compounds that, or the degradation of compounds that may be present through detritus and so on, those roles are carried out by microorganisms. We know that microorganisms also play an important role in aquatic and marine ecosystems. So what I've shown here are just 3 key insights from studies that have been done recently. The first study shows you that we are beginning to understand that all of us as humans have a core gut microbiome. So we all have a set of microorganisms that are present within our gut, and that depending on whether you may be obese or whether you may be lean, these microorganisms change, right? So this already shows you the key role that's played by microorganisms in our health status. The second study, which also used one of the approaches that I talked about earlier, used 60-ness or amplicon-based approaches to show that microbial communities differ depending on the level that you look at them from. So we know that microorganisms that may be found in parts of American soil differ substantially from those that are found in other systems. So microorganisms have very clear biogeographic trends, as we call it, and there are differences at local and global scales. So these differences are important because they determine the type of ecosystem services that we can derive from microorganisms. So work on the marine sphere has also shown the same level of importance of microorganisms in the global ocean. We know that microorganisms, as I mentioned earlier, play very important clear roles. And one of these roles is that if microorganisms somehow become disturbed, right, as a result of a stressor that may occur in marine environments, there are various examples of different stresses. We know as a result of the fossil fuels that we burn, the oceans are gradually becoming much more acidic. This has important consequences in terms of the ability of microorganisms and the food web structures that we observe in the oceans. A disturbance in something that you would not think about, like microorganisms, could impact the food that we derive from the oceans. So I'll talk a little bit about host-associated microbiomes. So we all have microorganisms in our guts. We know that when we're born, we essentially have a sterile environment in our mother's womb, depending on the publication that you read. And we know that as we progress through life, that our gut microbiomes change. You remember that I talked about how all of us have a core set of microorganisms that are present in our guts. We know, depending on whether you are breastfed or you use formula milk, the gut microbiomes tend to differ. We know whether you have solid food and whether you've used antibiotics. All of these things lead to differences in the composition of the microbes in your gut. Why is this important? Well, it's very important, as I've mentioned, because microorganisms are able to digest some compounds that your body cannot break down. So if, for instance, you are born or you are exposed to a diet that gives you a deficiency of certain types of microorganisms, it could determine your individual health outcomes and would have consequences for you downstream. So what is the status of microbiomics in Africa? Well, the situation here is not as glowing as the previous slide that I showed you. A search term using bacteria in Africa only reveals about 600 papers that were published in 2020. So compare 600 to 60,000 and you'll see that we are drastically behind in terms of what we have done in terms of understanding the microbiomes in African systems. And also on the right, you'll see that the fields that have applied microbiomics are substantially less compared to the multidisciplinary nature of microbiomics as applied in different systems. So what this slide demonstrates is that we clearly have lots of work to do. But I think that Africa has an untapped potential in terms of microbiomics. The fact that we don't know much about terrestrial, host-associated microbiomes, aquatic or marine ecosystems in itself represents a tremendous resource for us as Africans. I think studies on microbiomics, and I'll talk about how I see this later on, have the potential to become an important tool for fostering innovation across the African continent. So I'll talk about a few studies that we've chosen to focus on, and that are two projects that we are involved in. The first is looking at the effect of diet and geography on the gut microbiomes of African individuals. So in this study, what we did is looked at the gut microbiomes of people from a village in Venda and compared their gut microbiomes to people that are in urban settings. So using amplicons specific for bacteria and fungi, what you will see on the top part of the slide is that we see no difference in terms of the type of bacteria that are in their guts. But when we look at the fungal profiles, we see a clear distinction in the gut microbiomes of fungal taxa that are present within these individuals. So what does this actually mean? The fact that you've got differences in bacteria and fungi that are in the gut may not necessarily translate to anything that's meaningful. But it actually does, because at the bottom part of the slide, we actually looked at what are known as antibiotic resistance genes. Antibiotic resistance gene profiles revealed very clear differences between the resistomes, or the gut antibiotic resistance genes of urban individuals compared to those of rural individuals. This finding itself shows very clear health outcomes associated with the differences in the gut microbiomes of urban and rural individuals. We know that people in urban and rural settings have very clear differences in diet. And what these differences in diet and geography impose on the gut microbiome has very clear health effects. So one example that I could give you is that if we are taking processed food diets, that significantly changes the composition of the microbes that are inside us. When you look at medicine that's been developed, for instance, in people from Western countries, and you try and apply it to African individuals, the efficacy of that medication might not be as strong in these two individuals because their gut microbiomes or gut flora differs substantially. So an understanding of just how differences in the gut microbiomes change between these individuals is important because it allows us to tailor or go and develop a route of precision medicine that would allow us to have clear health-related benefits for African individuals. We've also, in our group, had a very strong focus on polar and marine environments. And there are several reasons for this, and I'll give you some of them in the next few slides. So South Africa has a very strong focus on strong geographic advantage. We, as a country, are surrounded by two marine environments and the Southern Ocean to the south of us, as you can see from this slide. So the Southern Ocean is the most important marine ecosystem. And we know, as Professor Erasmus said, that it actually plays a very important role in cycling nutrients and also in controlling the Earth's climate. Changes in the Southern Ocean may have very important consequences for the ecology of the globe. What I've shown there is part of what gives us a very strong geographic advantage is that South Africa has a base in Antarctica, and this allows us to carry out routine cruises that allow us to study the role of microbiomes in marine environments. So there are strong scientific justifications, as I mentioned, that the Southern Ocean and marine environments play an important role in sequestering carbon dioxide, and that through the actions of biological communities and microbial communities specifically, these marine environments microbiomes may act as potentially very significant feedback mechanisms that may make the effects of climate change worse than they actually are. So we know as well that part of our focus has been promoted by the fact that there's been very strong support in terms of promoting fundamental research that's focused on Antarctic ecosystems. This is something that I find I'm quite passionate about because as part of our obligations, we contribute towards global efforts through bodies such as the Scientific Committee for Antarctic Research, where we provide fundamental data on the functioning of ecosystems that are within African and South African waters. There are also very strong efforts to develop so-called global microbiome initiatives, and we as a country and our group has played a very important role in developing science policy plans on focus on what type of research priorities should be focused on as part of research in the global south, specifically in the South Atlantic. So I'll come back to this point as I conclude, I get to the point of concluding my talk, about how microbiomics can be a very important tool for innovation. I've spoken a lot about, during the talk, about some of the challenges that are associated with understanding microbial communities in various ecosystems. But I see microbiomes as key for improving health, food systems, and finally, economic prosperity on the African continent. We as Africans have a potential to leapfrog innovation by harnessing some of the insights that have been developed over the last 2 decades of research. We know, for instance, things that I talked about that have been challenges in terms of understanding microbial communities. There's been recent developments in terms of sequencing approaches, and other developments include innovations in terms of machine learning and other mechanisms that allow us to begin to study microbial communities and also their gene products in very complex ecosystems. Globally, there's been a strong boom in terms of biotech companies that have applied microbiomics for various services. But this boom has not actually happened within Africa. So what you can see on the slide there is just an example of the different soils that we have in South Africa. These soils may potentially hold microbes that have not been studied yet. So these microbes that have not been studied yet may potentially have very important gene products or tools. So things like CRISPR that are derived from microbial communities. Equally important developments can be found by harnessing the microbiomes of African ecosystems. So I see microbiomics and the development of microbiomics as a science as a potential important contributor to economic development and prosperity on the African continent. And being able to recognize the potential of microbiomes is one of the things that excites all of us and allows us to look past some of the despair that is currently happening in our country. So how will microbiomics at the University of Pretoria contribute towards the development of microbiomics? So we've got essentially 4 different pillars of research that are linked to 3 thematic areas of microbiomics. We are interested in looking at the microbiomes of natural and engineered systems. We are interested in expanding research on host-associated microbiomes. And we are interested in providing computational and bioinformatic solutions that can help other researchers also understand the microbiomes associated with our ecosystems. So I've put there some of the priorities that we see over the next 5 years, which will help us get to the point where we get to this economic prosperity that I talked about. So part of this is by establishing a strong national focal point on microbiomics. And this focus point will focus on using sequence-based analysis to provide insights on microbial communities in African systems. We see a very important niche in exploring microbiomes associated with natural environments and also with clinical systems. Over the next 5 years, we aim to aggressively pursue a research portfolio that's advanced by undertaking applied research, but also with a strong focus on providing fundamental insights. So hopefully in 5 years, when we check back on the development of microbiomics, you will come back to the University of Pretoria and see an internationally recognized world-class research entity with world-class researchers focused on microbiomics. And with that, I'll end my talk here. Thank you for listening. If you have any questions, by the way, please feel free to post them on the YouTube link. Thank you. Okay, so I've got a few questions. And the first question is, what advice do you have for young scientists that are interested in microbiomics? So maybe in answering that question, I'll go a long way around. So in order to excel as a scientist, the real exit degree for a science degree is essentially a PhD. So if you want to pursue a science career focused on microbiomics, you have to persevere to the point where you get your PhD. That will allow you to then choose the research that's of interest for you in terms of what you would like to study. And that's exactly how you make a contribution in terms of insights related to microbiomics. Hopefully that answers the first question. The second question is, what is the biggest gap in your knowledge of the microbiome for human health? Well, that's also it's a very big question, but there are various gaps. The first is that apart from the study that we did, where we looked at the differences in the gut microbiomes of people from Pretoria to those from Venda, there really hasn't been a lot of studies that have looked at the gut microbiomes of African individuals. So we know that there's been sporadic studies which have compared the gut microbiomes of people from rural parts of Africa to Western populations. But these studies are not really fair comparisons because we know that Africans have a very high source of genetic diversity, and genetic diversity has been linked specifically to differences in the gut. So that's potentially one strong limitation in our knowledge of host-associated microbiomes. And for that, we essentially need to study a lot more populations across the African continent. Okay, see, there's another question, and that is, what do you think will be the big breakthrough? So there's lots of potential breakthroughs that are just waiting to happen. So in the 80s, the discovery of Taq polymerase, which was discovered from a microorganism, came about as a result of fundamental research in different ecosystems. So the first part of making these breakthroughs are first to understand exactly what we have in our systems. So as I said, going into lots of different soils that have not been surveyed before and trying to understand what microbes are there. That is the first part of what will lead us to those breakthroughs. Once we know what microorganisms are there, then we can start exploring what type of gene products those microorganisms can produce. And in that lies unlimited possibilities. Things like CRISPR, which is widely used now in molecular biology, is one product that comes from microbial communities. When we look at genetic sequences from our studies, we know that a lot of the sequences that are there, we've got no idea of what they actually represent. And part of that exploration could unearth some of those big breakthroughs that could potentially happen. Okay, and then we have another question with regards to host-associated microbiomes. I'm just wondering why it is that the abundance of proteobacteria is seen in unhealthy humans compared to healthy ones. So this is also, it's not necessarily that there's an abundance of proteobacteria in healthy versus unhealthy. There are some lineages that are present throughout, but we know that things like proteobacteria could potentially hold groups that are linked to known pathogens. So E. coli is linked to gamma proteobacteria, for instance. So that could possibly explain why there's an abundance of those proteobacteria. But there are other proteobacteria that can be essentially benign and actually give good products that are not necessarily a threat to our health. Another question. I see that with microbiomics, the focus is mainly on bacteria than fungi. What do you think is the reason for that? So maybe in my talk, I've been biased for bacteria because they're not disgusting like fungi, but there is a lot of work that's done on fungi as well. I've talked about the one study where we looked at differences in the guts of African individuals. And this study shows that, and others that have been done, shows that there's a lot of work that's been done on fungi, but also on other microorganisms, including viruses. So there isn't a bias. The only bias is that I prefer to work on bacteria. Okay, what projects do you have in mind to do, but don't have time for? There's probably a lot of projects that we'd like to do, but it's not necessarily that we don't have time to do. It's more that we don't have money to do them. So we can find time somewhere. I'm not sure if there's another question. I see they're typing something. Okay, so the question is, I've spoken about humans, but what about animal health? So of course, this is also a very important area of research, and people are doing a lot of work trying to understand how things like the current agricultural practices, where animals are fed a lot of antibiotics, and how that contributes towards not only the health of the animals, but also ultimately to our health. So there's been a lot of work that's been done focused on looking at, for instance, antibiotic resistance genes and how they may be disseminated through humans and as a result of the different agricultural practices. So there's a lot of literature on that. If you email me, I'm happy to share some of those studies. So that's a complex question, which is the last one I have, which is the person asks, does taking probiotic make us healthier? The answer is probably yes, because there's been studies that have shown the beneficial outcomes of taking probiotics. But there's an interesting corollary to that as well, in that people that are essentially very healthy and not immunocompromised, there's no clear differences that are observed in some of these individuals as to whether they take probiotics or not. So if you are immunocompromised, probably taking probiotics will, of course, contribute towards making you healthier. And another question, is the microbiome perhaps a next frontier more significant than any biodiversity work done up to this point? Not just in terms of numbers, but an understudied core driver of ecology. I absolutely agree. If that was a question, I agree with your question. I think microbiomics is an essential driver for understanding not only the diversity of macroorganisms or larger organisms, because microbes play an important structuring role towards determining the diversity of plants, animals, and anything that's essentially on the planet. So I absolutely agree that it is the next frontier in terms of our understanding. And with that, I'm told that we've run out of time, but I'd like to thank you all for listening, and thanks a lot for all the questions that I received. And thanks again for everyone who's played a role in terms of organizing this and the production of the talk. Thank you.
Thank you very much, Professor Makhalanyane. What an interesting talk. It's a topic that popularly we know about in the context of probiotics and take them when you're on antibiotics, but there's so much more to it. It's clear to me that microbiomics are everywhere and we're really just scratching the surface. And I think that last question, elevating and saying this is not just a new knowledge frontier, but this is probably one of the most important discoveries and areas of research that wait for those of us in biology. And it's got tremendous, absolutely fundamental effects for human well-being, environmental health, and animal health. We all know that we're massively changing the environment around us. And so far, most of the environmental change work has been focused around the big drivers: land degradation, climate change, poverty, inequality, natural hazards, floods, droughts, and so on. But what Prof. Makhalanyane tells us is that there's a secondary knock-on effect which we know very little about. How do we affect the microbiome of the environment around us? And how does that microbiome affect our well-being when we initiate these drastic environmental changes? I'm also really happy about very impressed with the future view about microbiomics research at the University of Pretoria. And this is indeed an area that we would like to invest in the coming years. And I suggest that you watch this space and keep an eye on all our social media outlets for news and for new developments on microbiomics research in the Faculty of Natural and Agricultural Sciences at the University of Pretoria. I'm quite excited about the developments here, and maybe even before our next expert lecture, we can bring you up to speed. That brings us to the end of this installment of the NAS expert lecture. It's been really interesting. I think it's a great start to a series of lectures which I think will be no less cutting edge and will always have some extra food for thought for us. Thank you very much for joining us, and all the best. Stay safe.